Hi class, this is lecture 3, Regulation of Gene Expression. Here's an outline of the material that we'll cover in this lecture. For regulation of gene expression, we'll start off talking about promoters and promoter strength, and then move on to transcriptional regulation via inducible and repressible promoters. Then for expressing genes in heterologous systems, we will look at codon bias and then expression in eukaryotes. The most important thing to realize about the regulation of gene expression is the reason for it. Not all genes are necessary at all times. It takes energy and resources to produce molecules, transcribe RNA, translate proteins, etc. So if organisms were to translate all the genes in their genomes at all times, this would waste valuable resources and energy. As a result, it's important for organisms to regulate the expression of their genes. There are numerous methods for regulating gene expression. The most common is regulation at the transcriptional level. Generally, this is accomplished through two main methods. The first is a variation of the promoter strength. So organisms can have either strong or weak promoters. The second method is through the upregulation and downregulation of promoter activity, and the main way of accomplishing this is through the use of inducible or repressible promoters. So let's take a look first at promoter strength. Gene promoters can vary in their strength. There are strong and weak promoters. So you see in the diagram on the left a strong promoter. The strong promoter upstream of the gene induces a high level of transcription of that gene, meaning that many copies of RNA for that gene are produced. Because a large number of mRNA molecules are produced, a great deal of translation occurs from those molecules, thus producing a large amount of protein of that gene. A weak promoter, such as the one on the right, produces fewer transcripts of mRNA, resulting in a lower number of protein molecules being translated. The biochemical basis for differential promoter strength is the sequence recognition of the promoter by the RNA polymerase. So, for example, you have two regions on the RNA polymerase that will recognize specific sequences. The promoter sequence upstream of the gene contains two sequence regions called the -10 and -35 regions. I discussed these regions in lecture 0, so feel free to review that for more details. The sequences shown here on the diagram are the consensus sequences. That means that these are the sequences which are the most ideal for binding to the RNA polymerase. We'll look at variations from the consensus sequence in a bit. So the polymerase binds to the DNA upstream of the promoter and travels downstream until it reaches the promoter, at which point the complementary sequences between the polymerase and the -10 and -35 sequences form base pairs. This serves as the recognition sequence to bind the polymerase. After this, the promoter continues, polymerase continues in the 3' direction until it reaches the transcription start site and then begins producing the mRNA. The strength of the promoter can be reduced by changing either the -10 or -35 sequences or both. So these can be changed away from the consensus. So you see in the diagram each of the two sequences has two nucleotides that have been changed. So these no longer pair appropriately with what the RNA polymerase is looking for. So in this case, out of the 12 nucleotides in both regions combined, only 8 of those are the proper matches. And so this results in a weaker bond between the RNA polymerase and the promoter. And the issue here is that polymerases that reach the promoter and don't bind quite as strongly to the promoter sequence are more likely to dissociate and detach from the DNA molecule without actually beginning transcription. And so, in the end, fewer mRNA transcripts are produced, which naturally results, consequently, in fewer proteins being produced. The implication of that is that the deviation from the consensus sequence tends to be correlated to the level of transcription and therefore the amount of protein produced. So here we see a number of genes aligned around the -10 and -35 regions. You can see many of these genes have a variation from the -10 and -35 consensus sequences. Those are shown below. So from this set of genes listed here, only one, the TRP gene, has the consensus -35 sequence. All the others have some variation away from the consensus, and only two that are involved in rRNA have the consensus -10 sequence. So if we calculate the number of nucleotides which match the consensus sequences, we can see a clear correlation between the importance of a gene and the similarity to the consensus sequences. So of the 6 genes with similarities equal to or greater than 75%, 5 of the 6 are absolutely critical to viability. This includes 3 rRNA genes, 1 tRNA, and the gene responsible for the biosynthesis of the tryptophan amino acid. And so the criticality of these genes implies that the high sequence match to the consensus sequence is a result of evolution favoring sequence that will provide robust transcription of these important genes. Okay, so now that we've discussed promoter strength, let's look at the regulation of gene expression through transcriptional regulation via inducible and repressible promoters. An inducible gene is one that is induced by a regulatory chemical, and so when the chemical is not present in the environment, transcription of that particular gene doesn't occur. And so when the regulatory chemical is present in the environment, that triggers transcription of that particular gene to occur. The lack of transcription without the chemical and the induction of transcription with the chemical can occur via 2 different mechanisms, which we'll discuss in the upcoming slides. Similarly, a repressible gene is one that is repressed by a regulatory chemical. So when the chemical is not present in the environment, transcription occurs. So this is sort of its default setting. It's to be making mRNA transcripts. And then when the chemical is added to the environment, the transcription will stop. This too can occur through 2 different mechanisms, which we'll discuss now in the upcoming slides. One of the methods by which a promoter can make a gene inducible is through the use of a repressor protein. The repressor binds to the promoter, thereby blocking access to it and preventing transcription from occurring. When the inducing chemical is added to the environment, the repressor will undergo a conformational change, and that causes the repressor to detach from the DNA. That allows the RNA polymerase to access the DNA and start the transcription process, thereby producing mRNA molecules. The other method by which you can have an inducible gene is through the use of an activator protein. In its native default state, the activator is not able to bind to the promoter. When the inducing chemical is added to the environment, the inducer will bind to the activator, which causes a conformational change that will allow the activator to then bind to the promoter. That activator helps to recruit the RNA polymerase and have it bind to the promoter location where it needs to be in order to start the transcription process. So when we're talking about repressible genes, we have two analogous methods to the ones we just saw. You can have a repressible gene via a repressor protein. In its native default state, the repressor protein is not able to bind to the promoter, and therefore the RNA polymerase can access the promoter and start transcription. When the regulatory chemical is added to the environment, it will bind to the repressor protein. Because both of these are required, the repressor and the regulatory chemical, we refer to that as the co-repressor. So when the co-repressor binds to the repressor, that causes a similar conformational change that then actually allows it to bind to the DNA. So this is the opposite version of what we saw with the inducible promoter protein. And once that repressor has bound to the promoter, so naturally we have the fourth version of this, which is a repressible gene with an activator protein. And so in its native default state, the activator can bind to the promoter, and so that helps recruit the RNA polymerase to the proper location and allows transcription to occur. When the inducing chemical, which in this case we call the inhibitor, binds to the activator protein, that causes a conformational change in the activator. One prototypical inducible system is seen in the regulation of the bacterial lac-C gene. Uh, which is responsible for the uh, uh, production of beta galactose. So the lac operon consists of a number of genes. For our purposes here, only two are really relevant. They are the LAC-I gene and the LAC-Z gene. So the LAC-I gene encodes the LAC-I repressor, which binds to a region of the promoter called the operator. You see that in the diagram labeled with the O. And so LAC-I forms a tetramer that is the repressor protein. When lactose is present in the environment, it binds to the repressor and causes a conformational change in the protein that causes it to detach from the operator. Then with the promoter no longer blocked by the bound repressor protein, transcription of the beta-galactosidase LAC-Z gene and the other. So now let's take a closer look at how exactly the LAC operates. You can see here a more detailed molecular diagram of a LAC repressor binding to the promoter. You can see on the loop are the -10 and -35 regions. The fact that they are located in the loop means that the RNA polymerase is unable to access these. So now let's take a look at expressing genes in heterologous systems. We'll start with an important consideration. Here's the codon table. I'm sure you've all seen this before. We won't go into how to read this, but review your molecular biology text if you think you need a refresher on this. But the important thing to remember about the codon table is that most of the amino acids are coded for by several different codons. So as you can see for glycine, GGG, GGC, GGA, and GGG all code for glycine. Since there are 4 different options, you might expect that 25% of the codons for glycine would be GGT, 25% would be GGG, etc. However, this is not the case. The actual representation of the codons are not distributed evenly. As you can see from this table, the GGT codon accounts for 63% of the codons encoding glycine. The GGC codon accounts for nearly the entire remainder of the frequency of glycine codons. The important thing about these distributions is that it's not just the 3 nucleotides that make up the codon on the template strand. The organism typically produces tRNAs in approximately these ratios as well. That means that out of the tRNA molecules, in this case in the E. coli cell, only about 1% of those correspond to the codon GGA, and about 1% correspond to the codon GGG. And so I compiled this table to illustrate the differences in codon bias from numerous bacterial species. You can see E. coli, Bacillus subtilis, Mycobacterium tuberculosis, Salmonella enterica, Bacillus anthracis, and Yersinia pestis. The data in this table are by highlighting the most frequent codon for each of these amino acids in each of these species. We can easily see some differences in the preferred codon for these amino acids. For example, the amino acid alanine is encoded by GCT in most of these species. However, in Mycobacterium tuberculosis, GCT accounts for only 8% of all the alanine codons. Instead, the most frequent alanine codon in Mycobacterium tuberculosis is GCC. The difference is even more obvious for the amino acid serine. In all of these species, TCT is the preferred codon, while in Mycobacterium tuberculosis, TCT accounts for only 2%. Instead, TCG is the preferred serine codon in Mycobacterium tuberculosis. So this is more than a trivial point of interest. The codon bias that is displayed by different organisms can have implications on cloning and expression in heterologous systems. And further, codon bias is not only a factor in bacterial species. We talked about the LAC-Z gene, which encodes beta-galactosidase. Here we see the amino acid sequence of the LAC-Z gene. For the sake of our example, we don't need to examine the entire sequence. So let's take a look at the beginning part, the first 120 amino acids. Let's look at the codon bias involved in the amino acid proline. So this particular sequence has 15 proline So we see in this table the E. coli most common proline codon is in humans. However, the frequencies are the opposite. CCC accounts for 33% of proline codons, while CCG accounts for only 11%. The implications of the difference in tRNA frequency are that when we try to express this protein in human cells, any of the CCG codons will be translated with lower efficiency. This is because the tRNA molecules that recognize the CCG codon are approximately 6 times less frequent in human cells than they are in E. coli cells. So Consequently, during translation, the ribosome must wait at the CCG codon until an appropriate tRNA molecule enters the ribosome and can attach its amino acid. So rather than making the ribosome wait around for the proper tRNA, we can actually correct for this when we take a gene and put it in a system with a different codon bias. So we can do that by simply changing the DNA sequence of the gene that we want to express to better match the codon bias of the host. We do that by changing those codons that are infrequently used to codons that are more commonly used. So here we return to the first part of the E. coli LAC-Z gene sequence. And again, we have the CCG codons that are most common in E. coli, highlighted in the dark blue. And so if we wanted to express the E. coli gene in a human cell line, for example, the CCG codon is the least frequent, and so that would be the least efficient. So what we can do is change it to be one of the more efficient ones. So for example, the CCC codon, which is the 33% most frequently used codon for proline in humans. And so we see that here. All that's been done in the sequence shown on this slide is that we've changed the last nucleotide of the CCG codons to be a C. So now the CCC codon replaces what was previously the CCGs, and so the DNA sequence has changed slightly, but the protein sequence is exactly the same. So even though we took the gene from E. coli and then modified it just a little bit to work better in human cells, the protein that will be expressed will be exactly the same. It, of course, will be expressed with a much higher efficiency because the ribosome that's translating won't have to wait around for the less common CCG tRNA molecules. It can go with the more common ones that recognize the CCC codon. Okay, so that's codon bias. The example that we used in there was expressing genes in human cells. And there are some very important considerations to look at when we are expressing genes and proteins in eukaryotes. So let's go ahead and take a look at that now. So in prokaryotes, the coding sequences are directly transcribed in their entirety into mRNA, and then that mRNA is then translated into proteins. And so the coding sequence starts at the start codon, usually AUG, and then continues in these 3 base pair chunks all the way to the stop codon at the end. And so that entire sequence from beginning to end is transcribed into mRNA, and then that mRNA forms the instructions for the ribosome to translate into the proteins. In eukaryotes, however, there are introns, which are introgenic regions that interrupt the sequence. The remaining part, the segments that are actually expressed, are called exons. So the introns don't actually code for any amino acids. So when transcription occurs, the primary RNA transcript that is created still has these introns within there, and because they don't actually code for amino acids, not any that should be in the final protein, these need to be removed. And so the eukaryotic cells have splicing enzymes which will remove these from the mRNA. Uh, and after this processing step, the mRNA is referred to as mature, which means that it's had these introns already spliced out. And then from there, that mature RNA now has the proper coding sequence that is uninterrupted. And so now from beginning to end, this mRNA codes for what ultimately goes into the final protein. So in the last section on codon bias, we talked about taking a bacterial gene and expressing it in a eukaryotic cell. Here, the reason we're talking about introns is because very often in our cloning experiments, where we want to express eukaryotic genes, we choose to do so in prokaryotic organisms, usually bacteria such as E. coli, simply because they're so much easier to work with. They have much greater doubling times, or much lower doubling times. They have a much greater doubling frequency. And so overall, bacteria tend to be a much easier and better system to do experimental biology work such as this. And so that's why we do that. But if we are going to take a eukaryotic gene which has these introns in the DNA sequence and try to express that in bacteria, bacteria don't actually have those enzymes that can splice out the introns. So those introns would end up actually being converted into amino acid sequence that would then be interrupting the protein and would end up destroying its actual intended function. So how can we go about correcting for this? What we do is we harvest the mature mRNA from the eukaryote. So here in the diagram, we'd be looking at the molecule after the processing step. And then we take that mRNA and make a DNA copy of that, which then we can put into the bacterial cells and express. So I just talked about making a DNA copy from mRNA. That's not something that we had discussed previously. So very quickly, let's just revisit the central dogma slide, and then we will update it at the end. So of course you have your gene. DNA replication is making more DNA copies of that gene. Transcription is producing the mRNA copy of that gene, and then translation is making a polypeptide from that mRNA. And previously, this information flow was always thought to be unidirectional. So you went from DNA through transcription to mRNA, and from that through translation to proteins. As it turns out, there is another sort of bypass to this, in which you can make DNA from an mRNA template. In the natural world, this occurs in retroviruses, and so they encode an enzyme called reverse transcriptase. And so, as it would imply from the name, it goes, or it induces reverse transcription. which is going the opposite way as transcription. And so this is currently what the central dogma understanding is right now. As far as we know, we have not yet discovered any sort of reverse translation mechanism wherein we can go from protein back to any sort of nucleic acid. But we also didn't believe reverse transcription was a thing previously, so it's not entirely out of the question. It could exist and simply not have been discovered yet, but to our knowledge currently it doesn't exist. So in biotechnology, we like to take existing systems and enzymes and put them into interesting uses. And so, for example, with PCR, the standard PCR, we were able to take DNA polymerase and apply it in such a way that we could take a template and make additional copies of that. We've basically done the same thing with reverse transcriptase, and we call that process reverse transcriptase PCR or RT-PCR. And if you recall, I talked about real-time PCR back in the PCR lecture, also known as qPCR for quantitative, and I tried to stress that you should not refer to real-time PCR as RT-PCR, because that in fact is the acronym for reverse transcriptase PCR. So now let's go ahead and talk about that. RT-PCR uses the reverse transcriptase enzyme and a mature RNA, mRNA as the template, and through the process of RT-PCR makes complementary DNA or cDNA, and we call that complementary in that it's complementary to the mRNA that served as the template. So here in the diagram we have an mRNA. This would be the mature mRNA, and so it no longer contains the introns that were in the DNA sequence. Most eukaryotic mRNAs have this poly A tail at the end, which is just a string of A's. The nice thing for the purposes of RT-PCR is that this poly A tail we know the sequence of. It's just a bunch of A's, but it's always at one end and serves as a segment of known sequence. So what we can do with that knowledge is create a primer that will bind to that sequence, and because it's all A's, our primer can consist of all T's. So we take our poly T primer and bind that to the poly A tail, and then from there we can add reverse transcriptase, and it will begin forming a strand of DNA bound to the mRNA template. And when the DNA synthesis continues, it will fill in the rest of that molecule. And this molecule here is the last one we saw on the previous slide. I simply didn't have enough slide space to continue going down. But from this step, what we would do is add an enzyme that is an RNase. Often we'll use RNase H. What that does, not surprisingly from the name, is it will digest the RNA strand that's forming the RNA DNA hybrid double strand, and so once that RNA has been fully removed, we'll add DNA polymerase such as a Taq that will form the other strand of the DNA, and this part is essentially just regular PCR. And what we end up with as our final product is a double-stranded cDNA that is a DNA copy of the mature mRNA that we started with as our RT-PCR template. And once you have completed this, then you can take this cDNA and then use that and insert that into one of your cloning vectors, and then it's suitable for expression or any other purposes. Okay, here are the references for this lecture, and I hope you found that useful, and don't hesitate to post any questions on the website if there was something that wasn't entirely clear, and I hope you guys have a great week. Thank you.